Everyone, welcome to London. Welcome to London Calling 2025. And we, the Nanopore Innovation Team, are going to talk to you about how we're going to take all of these really big dreams that we've had for many years together as a community and turn them into real possibilities with some practical yet ambitious considerations. Word of caution, we will be making some forward-looking statements. I will quiz you on the contents of this slide after the talk. It's really important for us to begin with everyone who's in this room, everyone who's online, and really say that, there's nothing happens in this organization, nothing happens in this company, nothing happens in R&D without deep consideration for the community, for you, our community. Think, do, act, how you really want to interact with our product. And that's what brings it all together. So the tech talk, why do we do it? And that is a question that we ask ourselves increasingly, like the 2 weeks before. Why? Why? It's a lot of work. But whenever we ask ourselves, why do we do it, we always come back with a few really, really simple reasons. This is what I like to call an ecosystem between Nanopore and the community, the industry. And it's really important, we find it really important to make sure that you understand the context of our products, what we're doing, why we're doing to them, where we are today, and where we want to go in the future. You've got a lot of decisions to make every day of the week as you point your direction into in a certain way. And what we want to be doing is helping you make those decisions by being as open as we can with where we plan to take our technology. The other reason we feel that it's really important to keep everyone up to date is because we are dealing with a platform that has immense headroom. So we have one platform, one engine that drives it, and we can place this platform into any form factor, and we can adapt the platform to read any biology. And it takes real team effort to bring all of this together, and we thought we'd give you a little preview of who our team leads are back at base at Oxford Nanopore for R&D. Quite a few of them are in the room today. We've racked up about 150 years of Nanopore experience, and that's not me. So it's shared between the groups, and over 2,500 patents. All of these guys are powered by exceptional talent at Nanopore. It's our 20th birthday this year. So we always say it's taken 10 years to took the first 10 years to make something that no one really thought could work, and then the next 10 years to make it, to bring it to where it is today. We're powered by the exceptional talent of the community, and of course we're powered by the broader genomics industry that drive all of our ambitions forward. We've proven over the years that we can deliver on innovation. Every year, new exciting technologies come out, and we really try and pack as much of the platform improvements through chemistry and software so that you, our user, our community, can invest in one instrument and make sure that that instrument keeps returning year after year, keeps returning with better and better performance. Through all of these releases, we've now ended up in a really stable position for our Q20 chemistry that we launched a couple of years ago now. So we've tidied up all of our product roadmaps. We've got the MinKNOW flow cells doing R1041, RNA. We have the Q-line R9 released and supported for many years to come. PromethION, and of course all of our kits have moved mostly into the release for the Kit 14 chemistry. Many of you know that at the start of this year, we did a little bit of a rebasing of our product range, really make sure that we're focusing on the right things. So one of the things we did early this year is move Flongle into an available upon request states, so that we can really focus on MinKNOW and PromethION and making sure that these are delivering for you. We also moved to a CapEx first model, And that was really to start fitting in with more industry standard practices, making it easier for everyone to do business with us. And despite that, we're still happy to say that we are the most affordable and accessible technology on the market. So with that introduction done, we're going to get into the main meat of the talk. And there's going to be a lot of content in here. But I wanted to make sure everyone could walk away with sort of five key messages from us, which is that we're committing to deliver the most complete biology. We're committed to making that biology easy to use. We are going to keep driving affordability and scale. And we're going to support the transition of this technology into the applied markets to accelerate everyone's impact in this room. And then we're also going to really unlock Multiomics to power your breakthroughs and make sure that, that this venue is full for many years to come of amazing science. So let's get started. Generating the most complete biology. It's quite hard to sort of try and capture all of the different angles that nanopore data can. I knew my mum would call halfway through. She always does this. She's got the best timing. I'm sorry. So generating the most complete biology. There's a great saying from Galileo, which is, measure what's measurable and make measurable what is not. So this, after many weeks of debating, this is where our teams have landed about the new dimension of nanopore data that we can deliver. So we really care about genome mappability. We care about structural variations, SNVs. So we care about variant detection. We care about methylation. And then we also think phasing is exceptionally important. And, of course, genome assembly. So we really try and cover all of these bases when we move the technology forward. So in order to really unravel all of this, it needs to be easy to use. And so our machine learning team, they work a lot on a product that's called Dorado. Many of you will have heard of this before. It's mostly deployed in MinKNOW, but it is also available standalone. And what it does is it takes core analytes, be they sort of DNA, RNA, or sort of novel methodologies. It ingests that data from our sequencers. And a lot of people think Dorado is a base caller, but it really isn't. It's an architecture that houses the ability to unravel that raw signal into biology. So it does house our canonical base callers, our modification callers, variant callers, aligners, barcoders, and we'll be doing polishers too. That then feeds into Epitome for bioinformatics or any other tool that you've developed. So on canonical base calling, our teams have continued to deliver performance enhancements. So we really focus a lot internally on our hack model, our optimized base caller that runs in real time with all of our sequencers. And the teams are not only worried about pushing the raw read accuracy forward, but really about getting into the signal and improving areas of the base caller that can have a really big impact on things like SNP calling and indel calling. And so with these latest improvements, pushing simplex into Q22, we're now seeing a really big enhancement in indel performance. Now, it's also quite important for us to start taking more ownership of variant callers. We have a lot of you that are coming to us for, one little minor thing. Dorado's out now, so anyone who wants to start playing with this, you can do that now. Our teams also spend a lot of time now moving beyond the base caller into the variant detection stage. So what we'll be doing is starting to integrate variant callers with Dorado. We have an alpha version of that in Dorado v1. And again, we're starting to see improved indel calling versus generic tools. And this is really because our team are really pushing that optimization towards nanopore data. A lot of the variant callers that are out there for long reads are generic, and we really want to try and get it optimized and honed for nanopore data. There's also a growing amount of interest right now in somatic variant calling. There's a lot of panel companies out there, a lot of oncology panels, and we know that we've been quite good, actually, at doing low-frequency variant detection, but it is really, really difficult to validate. So we have got a tool in development to do this. We can get to VAF levels below 5%. And if you're interested in this, please do reach out to the team if this is something that's relevant for you, because we do want to work with as many different types of applications as possible to really start more generalizing this capability. So moving from variant callers into modifications, the first thing we have to do with modifications is actually teach our canonical base callers that modifications exist. And through all of this activity, through all of this effort, our teams have been able to massively enhance the consensus of things like bacterial calling, so that you now get Q60 nanopore-only bacterial genomes. All of these models are in the upcoming MinKNOW release with supported end-to-end workflows. And all that we've done in order to drive that performance forward is teach canonical base callers that modifications exist. And I think that was a really interesting part of the first talk today from NIHR around how some of the modifications that they see in their saliva samples might be impacting this. And so again, it's really good for us to be working with you, the community, so we can drive this not just from bacterial, but into human and other organisms. So now that we've taught our taught our base callers that there are modifications present, the other thing we want to do is actually detect them. So on that front, we have an unparalleled offering. So we simply capture the beautiful complexity of nature. Over 10 different modifications are detected by the nanopore system today. And of course, as we add training and add that capability, more and more will come through. The teams not only add the ability to detect modifications, but they really push the single raw read accuracy of modification detection forward. And each and every community meeting, you'll see a really, really big enhancement in the performance of all of our modification detection. So from natural modifications, there's a really interesting application coming out around synthetic modifications. So some of our apps team have spoken about this before. There are quite a few there's quite a lot of publications out there around this as well. This is where you can paint 6-MA into the genome through methyltransferase and really start to access sort of chromatin stenciling, putting a new lens on the mechanisms of gene regulation. It's a really, really exciting application. When it first came out, we actually needed to have a different base caller model, but since then we've actually taught the base callers that this application is happening. And so again, with the latest base callers that are going out in the next version of MinKNOW, all of this has been fixed. So in one simple experiment, you can get your variant calling, your chromatin accessibility, and you can also get native methylation. The mod kit, which we use to do the analysis, is available now, so anyone who's interested in this application can absolutely go for it. And then this one, someone told me to look out for the raised eyebrow look. It's apparently a little contentious, this one. Modifications. We want to try exploiting or exploring the signal for different things. And one of the things that it might be really helpful for is phasing. Oh, someone said yes. Okay. There'll be debates later, but we're excited about it. We think it's interesting, and we really want to start a community conversation about this. And so what we see here is sort of conventional phasing methods and enhanced methylation phasing. We have a new tool out, POMFRET. It's a fish, if you need to Google it. I've never eaten one of them, but I'm sure they're delicious. So it's methylation-aware phasing. It's an implementation of sort of prior concepts for metaphaser. I think I've seen Fritz around today somewhere, but it's something that the teams have worked on together with that. Finally, onto assembly. So assembly is it's moved really, really fast over the last few years. And since the dawn of the T2T, we have, of course, our T2T bundle. But then there was an amazing paper published earlier this year, High-throughput ONT, which is a tool that actually optimizes assembly so that you can use what we call probably normal read lengths, 30 kb, or long, not ultra long, but you can use 30 kb read lengths, which allows you to really scale your projects because the sample prep is far simpler. And you're getting assemblies with a sort of 100 to 130 megabase contigs at a Q-score of Q47. So we really, and also really reducing the compute needed to assemble a genome. So that's an amazing paper that's come out in the last 2 to 3 months, and is really scaling and accelerating the rate at which you can do T2T genomes. Now, all of this is all coming in and bundling into Dorado. Beyond that, we have our Epitome product. This is so that we sequence base call everything with MinKNOW. We can then analyze all of your data with pre-configured workflows in Epitome. We can integrate those workflows into your own pipelines, or you can also get compatibility with tertiary analysis software providers. So the team have carried on improving the user interface. It's a nice new one with 20 pre-configured analyses. But we've also been working a lot on the Epitome server so that it can really integrate not just with our instruments, but also all of your infrastructure. Quite important as well is the team are working towards regulatory compliance on Epitome so that you can use it more comfortably in applied settings. Finally, MinKNOW, the engine that runs it all. These are the release timelines for MinKNOW. So we will have in the next release in May time, all the Dorado 5 models, Blackwell support, and starting to move towards Ubuntu Focal and Noble support. In Q3, something quite important is happening to MinKNOW, which is that raw data output will be off by default. So, of course, we'll be messaging everyone to remind them. But we feel that as the data has moved on and the quality has moved on to such a level, and everything is so rich, that the raw data should now be a switch on as opposed to switch off mode. So we started this section talking about all the different multimodal availabilities of nanopore data, and this is how our team have managed to plot all of those along one continuum. So our gap-free mapping coverage, this is human genomes, so 99.1. We've got fantastic SNP, SV, coding indel scores. The total number of DNA and RNA mods is now at 12, and you can see all of the other pieces as well. So hopefully you'll feel, and you can see that it's a very, very complete data offering from Oxford Nanopore. But again, we're working hard to solve the informatics piece. Next, of course, comes the wet lab, or the whole entire end-to-end. So for 2 to 3 years now, we've had a really big focus on making this biology more accessible. We focus on a few key areas, like so research, clinical, applied, industrial, and with our teams, with our customers, with the whole community, we start bundling up these end-to-end workflows to make it a lot easier for everyone to get up and running and get successful outcomes. We've had small genome workflows for a while. There are a few enhancements that are coming that I'll talk about shortly. But again, here you can go anything from targeted applications all the way up to full genomes, metagenomics, and SynBio. And there's an enormous amount of energy and opportunity in this, be it customers doing it directly, being service providers starting to offer it as a service. I think everyone sort of knows the Plasmidsaurus logo really, really well now, and I know a few of them are in the room today, and they've really helped take plasmid sequencing to the next level. So key upcoming improvements in small genome workflow is the release of a non-fragmenting rapid chemistry. This is something we hear a lot in terms of demand. People want to be able to sequence amplicons without the fragmentation of the rapid chemistry we have today. And so these chemistries, these primers, they are optimized assay by assay. So the first one that we're going to be launching is 16S with ITS and fungal primers. a 24-multiplex kit. As we get this chemistry right, we think it will enable amplicon QC testing, and versus sort of current methods, there will be no upper sequence limit, no primers needed for sequencing these, and you should get a really rapid turnaround time. The teams, thank you. Everyone's happy. The teams are really working on 384 barcodes. I know we've said this for many years, but we are really committed to this. It's been a really fantastic journey that we've had in this space, and we'll be working with our partners to make sure we do this right. So moving from small genomes to human workflows, we've shown variations of this slide for a while. It actually updates every few months. So we have the ability to do targeted sequencing, low pass, standard 30x, the assembly. No one's really sort of come up with a new word for an assembly which isn't a T-to-T assembly, but is a really good assembly. So again, I think more conversation for dinner. And then, of course, you've got the T-to-T multimode. So for each and every grade of discovery around the human genetic profile, we have a really strong offering. We're going to talk about targeted for a little while, because it's something that we often forget to do. We get excited about other things. Last year was all about T-to-T. And so targeted, actually, Nanopore has some really, really good, some fantastic features. The fact that you can do amplicon panels, hybrid capture, adaptive sampling, things only Nanopore can do. And you can go from really small, tiny fragments to much larger fragments. You can resolve complex genes of interest, and you have an enormous amount of flexibility. And there's quite a number of applications that are coming out now. So if you think of things like amplicon panels, library prep from PCR is 15 minutes. You're sequencing amplicons from 300-400 bases all the way to 12 kb. You can run flow cells, stop them, wash them, reload. You have all of that flexibility, which gives you richer insights and really, really fast turnaround times. And working with our partners at Asuragen, they've developed a carrier screening panel that characterizes 10 genes, 6 of which are really challenging to read with short reads. And so this single panel is going to increase the number of disorders that these PCR panels can screen, but also replace 5 alternative technologies with one single test. And so I think a lot of people always think of Nanopore as a long-read sequencing technology, but it is actually a sequencing technology, and it's fast, and it gives you richer insights. Same thing on hybrid capture. Again, we can read much longer reads. We've had a collaboration with Twist now for a number of months. And what we have, as we've developed our platform's becoming compatible with commercially available hybrid capture panels. So again, sequencing any target size, you can do instant library QC. We've had some of the FASs over at our facility, and as soon as they see the live alignments in MinKNOW, their eyes light up with joy. And again, richer insights, affordable, no batch penalty, and you can get panels, 49 gene panels, 2 megabases, resolving complex genes like CYP2D6, getting phasing into all of these, and it's highly, highly scalable. There's also lots of other really interesting targeted methods. We had a really great start to the show last year with Carol Greider, who talked to us about telomere sequencing and how actually some of the discoveries, since they've started to utilize things like nanopore technologies, that each telomere is regulated around its own unique length. But of course, you really need to be able to scale this to start doing volume-based experiments. And so we've been working with a team to make sure we pull a protocol together that enables between 1 and 12 samples to be sequenced in a single run, full analysis pipeline and epitome, and that protocol is out now. So then moving to adaptive sampling, the Nanopore unique way of targeted sequencing. For those of you who've never seen this before, you upload a reference into MinKNOW. As the strand approaches, MinKNOW, the Nanopore starts reading it in real time. If it's a region of interest, it sequences it. If it's not region of interest, it rejects the read. What you get out of adaptive sampling is a really unique mix, because you get your enriched genes, but you also get a low-pass whole genome sequence. And you can use that low-pass to look at things like large chromosomal gains or losses. You can use that for polygenic risk scores. So you get an enormous amount of biology and a huge flexibility to design your panels when you use adaptive sampling. A lot of people think adaptive sampling is something that a few people run. We actually get, it's one of the biggest reasons that, that we get customers coming to talk to the Nanopore group. And the teams have been working tirelessly, our software group's been working absolutely tirelessly on making core system improvements. So we've improved the response time, so the enrichment's better. Our software can run full P24 devices, all systems go. We've launched in-depth how-to guides. The entire product has become far, far more mature than where it started. We've also launched a run-until, and we've paired this with Nomis. And so this actually allows the system, you can tell the system what coverage you want of each genome. And when that coverage is reached for that particular genome of interest, it stops sequencing it and starts accelerating the other genomes. So you can get a massive reduction in time to results by actually being far, far more adaptive with them. And it's really getting quite popular. I think, Matt Luce, has your name been said 5 times now on stage? You want another one, Matt Luce? Okay. It's been really, really exciting. Of course, sort of the work that Matt's done around this, adaptive sampling wouldn't be where it is without all of his, don't break your glass. It wouldn't be where it is today without an enormous amount of dedication and energy from that team. But we're also getting an enormous amount of other interests from oncology panels, and the teams are working on a hereditary cancer panel that will be going out later this year as well. So we've spoken about targeted. Now it's over to the human workflows, the whole genome workflows. We have been working with a very large number of you for a number of years, learning what it takes to sequence genomes at scale, so going to 100,000 genomes in the Emirati Genome Program, 1,000 genomes you heard from the NIHR group earlier today. That's been fantastic. And I think Kimberly's here as well in the audience from the NIH CARD group. We have the workflows that we need for this. So the preparation, sequence analysis workflows. We have T2T workflows. We can scale with 30x workflows. And we're creating a lot of low-pass workflows so that customers can complete existing data sets with epigenetic and structural variation. On T2T, the full bundle that we launched last year, it is available in the shop, but you have to scan a code in order to get there. This is because we want our field support unit to be in touch with everyone who wants to be doing full T2T. We want to make sure that you get a really, really good experience out of the box. So if you're interested in it, please scan this code, and you'll be given access to it in the store. The method in practice, our apps team sequenced a Puerto Rican genome, 56x coverage, POR-C, and an automated assembly delivered 34 out of 36 complete T2T contigs and 46 out of 46 raw T2T contigs. So it's no longer 23andMe. We should now be talking about 46andMe. And this, again, is all down to the work that the community has been doing to drive this science forward. So then we talk about our 30x capability. We've shown this slide, I think, a few times before. We have a high-throughput production workflow. You heard some of the details of this from the NIHR group. When you're running this with high-quality blood samples, customers are getting over 90% of their runs delivering 100 gigs, averaging about 120, 130 output. Now, this is a slide that we've shown a few times before, and really what it's saying is that we have a very, very clear path on how we're going to reduce the cost per genome. And the prevailing question that I get when we sit inside any meeting room is, are we there yet? is a prevailing question. And actually, I've been talking to people who, I don't have kids, but I'm talking to people with kids like, well, does this still happen despite the fact that there are sat-navs? Apparently, apparently they now say you've got 17 minutes left. There's now 16 minutes left. So yeah, it's a thing that will never happen. That will never stop happening. Sorry. Anyway, what's taking so long? When we launched R10, we as a company we had far less demand for our products. It was a long time ago. It was sort of early days of PromethION. And so we could launch a product, we could launch something for accuracy, and really worry about how you scale it, how you work on robustness. You could worry about that later. We can't do that now. The demand that we have on the PromethION range is really big. We're trying to launch a product which improves robustness, so it has to be robust. And what the teams have been doing is working really hard through the development to make sure that all of the raw materials that we're getting are actually consistent, so that we can deliver the performance that we want to deliver and that you deserve. We also need to consolidate releases and ease the transition. There's a lot of large programs happening, a lot of research studies, and that sort of start-stop, that needs to smooth out. So we need a couple of things to get this out the door. One of them is the ability to multiplex genomes onto flow cells, and that high-throughput barcoding system is in very late-stage development now. We take 96 barcoded libraries, you pool them, and you barcode pool them, and you can run 24 samples across 24 flow cells, or run 24 samples across fewer flow cells, so that you get to that 99% of genomes getting to 30x coverage. So the other enabler, of course, is higher output chemistries. And there are two things here. We've spoken about our ability to reduce pore blocking with an improved flow cell chemistry. You've seen that we've been working hard in development to make sure that all the raw materials are coming through at the standard that we want. It's the same nanopore, same base calling models. It removes the need for washing and reloading, and it improves the output for all libraries, including the long fragment ones. And that typically takes you from where we are today. It adds about a 30% improvement in output. The other thing the team are working on is an increased output with a new kit chemistry. And so that is generating more data per run. We're going to be targeting the launch initially at human high-throughput workflows, and after we've done that, after we've proven that we can get this chemistry good and stable in the field, we'll start the integration into other chemistries. When you put those two things together, we're targeting output improvements above 60%, close to 70%, and that's how we really push the envelope towards the 200 gigs per flow cell. So with that, we've landed with a simpler product range today, 2 core flow cells and instruments to run them on. We are investing in these products, be it on the support of getting users set up when they're running things like MinKNOW. So we're working on a companion laptop. There's one out in the live lounge so you can have a look. We'll be running a beta test of this to see how well it works for customers. We're investing in our PromethION flow cells. We're investing in our MinKNOW flow cells, simplifying everything, getting that consistency up, making sure you've got great product. These two products also have an immense ability to scale for the future. The AI revolutions here, there is no doubt that they will continue to drive compute, be it NVIDIA, be it Apple, be it ARM, that race is on. And that is a race that we benefit from. We've shown, and you can see Chris Seymour's talk tomorrow, we've shown that we can create larger models. We can get up to Q27, Q28. So please go and join the Data for Lunch presentation tomorrow to learn all about that. And we're also using AI to accelerate the development and engineering of future nanopores that we can use to further enhance our performance. With this product range, we still have 2 areas that are missing. We need to drive accessibility, decentralization in applied markets, and scale and cost. Around the democratization of sequencing, 3 very simple things. Lab-free, easy to use, sample to answer. And you always have to get a cat into your slides. So this is the reason actually many people join Nanopore. This is what they want to see. They want to see a completely decentralized instrument. They want to see sample prep, flow cell loading, sequence. The whole entire piece has to come together. And this is what we are doing with our SMIDgine ASIC program. We have a lot of sample prep technologies in-house. We've got fully automated manufacturing. We have shown that we can do integrated portable sequences before. We are bringing these together to design really for the applied market and get all of you, what many of you have really found inspirational about the capability in the future of this technology, which is a product that we could have one day have in a doctor's office. The next side of it is the ultra high throughput. We call this project GargantION last year. And of course, data output's really simple. It's the speed of the enzyme multiplied by the number of pores multiplied by time. We are working, so this technology maintains all of the data richness that we enjoy today, all of the simplicity of sample prep that you enjoy. It's based on that voltage sensor technology we've spoken about before, and an estimated output of probably about a genome per hour per flow cell. Scalable beyond this with faster chemistries and multi-flow cell instruments. So that is where we are driving our instrument and our product range to. So we're fully scaled today. We've got an exciting roadmap. And now to talk about biology is my partner in crime, Lakmal. Thank you.
So that was a fantastic overview of oh, thank you. That was a fantastic overview of the evolution of our platform, what we have done so far with the improving fidelity, the output, the modifications, assembly, and so on. So in this section, I'm going to shift focus slightly and try to talk about a new era of biological insight, how we can make multiomics modular. So like my intention is to show you what else we can do beyond DNA sequencing. So let's start with something familiar. So this is the iPhone, iPad, and the Apple Watch. So as you know, like this is one core technology, different devices, same set of applications, but used in different ways to cater different scale and different needs. So this is very similar to how Nanopore ecosystem works. So we have one platform, any form factor, and any biology. So let's start with something familiar to begin with, cDNA. So we want to capture longer transcripts as possible. So this is the central dogma of biology, DNA transcribed into RNA, and RNA translate into protein. So as you know, like this process is far from linear. There are multiple levels of regulations going on at each stage. But what I want to highlight is the transcription state, when a gene is transcribed into messenger RNA molecule, so introns are removed and exons are spliced together to make the mature RNA after splicing. So when this process happens, so exons can be spliced in multiple different ways, which means one gene can give rise to different isoforms of mRNA transcripts. So that's why it is very important, the ability to read full-length transcripts give rise to isoform diversity, alternative splicing events, gene regulation mechanisms, and they are often missed by short-read sequencing. So the ability to read the full-length transcripts is so important to get all this information correctly. So as you know, like we have a cDNA kit already in our commercial, so we offer the cDNA kit. And with this cDNA kit, what you see in the, on the right-hand side is the protocol. And in this protocol, we have gone through every single stage to dissect how we can improve this protocol to improve the read length of transcripts that we are producing. So our teams has figured out that the reverse transcription state, or the change in the polymerase enzyme, has the maximum impact on the read length of the RNA that is produced in this protocol. So they have changed that DNA polymerase enzyme, which is only a third-party reagent. It is still the same kit, same flow cell, nothing has changed, but it is a protocol update, and with that update, we have got a fantastic results improving the read length of our RNA transcripts. So we are getting about 70 million reads, and we are getting about 2.5 to 2.7 kb transcripts in a Promethean flow cell. So with that, we are also upgrading our barcoding kit for cDNA. So we are mainly changing that barcoding kit from plate format to, vial format to plate format to make it easier for high throughput analysis. So I'm showing some of the data from our new cDNA protocol improvement. So what I'm showing here on the left is this aligned read length. In gray, what you see is our current baseline protocol. Average read length is about like 1.3, 1.4 kb. In blue, what you see is the new protocol improvement. Dark blue is total RNA, and light blue is poly-A enriched RNA. So the N50 of that blue is 2.7 kb, which is a significant improvement, which means you now be able to get full-length transcripts when you do our cDNA analysis. So on the right-hand side bottom, you see the output improvement, the GB. Total output in blue is the new protocol. In gray is the old protocol, or the baseline protocol. So we get a significant improvement of total output. And on top right, what you see is the total number of reads. So with the new kit improvement, we are getting about 70 million reads from a Promethean flow cell. It is slightly less than previous baseline method. That is only because the read lengths are now much higher. So obviously, the total number of reads has to be lower. So just to show some data, so when we do generally cDNA analysis, you need to do experiments in duplicates, replicates. So what this shows on the on left is 2 replicates of increased median transcript length. So if you get full-length transcripts, it is towards the 100%. And you can see that compared to baseline, in the new protocol, you get very much to the top of the range of full-length transcripts. So the light blue is the median read length. Purple is the mean read length. You can see median read length is almost closer to 100%, which means you get full-length transcripts for most of the transcripts. In the middle plot, what you see is the correlation between the replicate 1 and replicate 2. So if we want to make sure that there are no bias, or there are no bias introduced in the protocol or the preparations, so correlation is really nice, which means when you do replicates, you get the gene counts right in all cases. On the right-hand side plot, what you see is the quantification. So we introduce IVT spike in transcripts to make sure that we get the right quantification. And as you can see, again, what you expect, what you see is the rep1 and rep, so in the x-axis is what you put into the flow cell, and what you get from the flow cell is on the y-axis. So correlation is really high as well. So you can accurately quantify how much transcripts you are getting as well. So again, to showcase the improvements between the baseline protocol and the new protocol, in gray, what you see is the baseline protocol. In blue is the latest protocol. And you can see this is all the transcripts mapped into a gene, 18 kb. And the full, you can see you can get all the full-length transcripts in our new protocol from 3' end to 5' end, or vice versa. So it's really nice improvement, which means you can now get full-length transcripts from our cDNA analysis. Not only the full-length transcripts on the top right, what you see is the poly-A tail length of cDNA. So from 140 to 40 length transcripts, and you can see poly-A tail length is the solid bar is what you should expect, and the graph tells you what you should get, and you can see the correlation is really high as well. On the bottom right, what you see is the capture of protein coding and long non-coding RNA, again, compared to gray and blue, current protocol versus new protocol, log count, you can see you can get more full-length transcripts from the new protocol. So this kit is now in beta testing program, and some of our collaborators are already testing this. So this data is coming from Professor Yasuhiro's lab, who spoke earlier about RNA and cDNA. So they use a completely different cell line, and in that cell line, their current protocol gives about 753 base N50. The new protocol on the right gives 1.9, almost 2 kb. So it's almost double the length of the cDNA. So this is not our data. This is coming from Professor Yasuhiro from Riken University. And many thanks to Dr. Kazuhiro as well for sending us this data on time. So moving to a single cell from cDNA. So as you know, like our protocols, our chemistry and the devices are compatible with 10x. So 10x has many different protocols. So our protocols are compatible with their next Gen 3 and next Gen both 3' and 5'. So their latest chemistry is Gen X. Again, from 3', 5', our protocols are compatible with them, and we have fully released end-to-end workflows with that. And then on spatial transcriptomics, Visium 1 is already released, and for analysis pipelines, SVP protocols, end-to-end protocols are already released, and fusion protocols are in development as well. So again, just to show why you need to do long read or ONT versus short read in cDNA in single cell. So on the plot on the right bottom, what you see is the at the top in light blue, you see the ONT protocol. At the bottom, what you see is short read protocol, and you can see clearly the difference, how much data you are missing in the transcripts analysis when you do short read cDNA analysis versus Oxford Nanopore cDNA analysis. When you do ONT single cell, you can see that you not only get the full-length transcripts, but you can quite nicely get the exon junctions as well. And on the top left, what you see is the gene counts between the short read and ONT protocols, and you can see the correlation is really high for this single cell data set. And on top right, what you see is the box plot for the gene expression. Again, correlation between ONT and short read sequencing is quite high. So moving on to direct RNA. So direct RNA is something, as you know, like only Nanopore can do. In every other technology, you have to convert RNA into cDNA before analyzing. By doing so, you lose all the native information. But with Nanopore, you can do direct RNA. You can move the DNA RNA strand directly through the Nanopore. So we have spoke about direct RNA sequencing last year. So we have released a new kit, which is RNA 004, which gives really nice accuracy. So you can see accuracy is almost Q20. And with that, we also included really nice features. You can see that the read length is about 1.8 kb, and accuracy is really high. And high output, you get about 25 million reads from a Promethean flow cell, and more importantly, you can get the poly-A tail measurements. So you can see on the plot, like the solid line is what you expect, and the graph shows what you are getting. So it's really nicely correlate. You can very accurately measure poly-A tail length from mRNA transcripts, and also we can do short read previously, with the previous kit, we could only do about 200 nucleotides direct RNA sequencing, but with the new protocol, we can do anything above 50, and that actually enables doing quite a lot of interesting biology. So you can see with the short RNA on the graph on left, it's a baseline. So the output is about 120, and this is from 90 nucleotide transcripts. And on right, what you see is tRNA. So because we can do short-read direct RNA sequencing, it enables tRNA sequencing as well. So tRNA is a big field now. There are no many methods that you can do proper tRNA direct analysis of the molecule directly. So on the plot at the bottom, what you see is E. coli tRNA. So there are about like 20 different tRNA in E. coli, and you can see all the tRNA in E. coli. We can see nicely, not only like we can detect them, but also we can get the relative quantification of them as well. So one thing that we are doing recently is introducing a new barcoding kit for direct RNA. So many of you, like when we talk to RNA community, so you kept asking when do we get barcoding kit, because we understand that it is important, it brings the cost down. So we are now introducing our new direct RNA barcoding kit. So this is already in beta testing, and it is the same motor protein as RNA-004, same flow cell as RNA-004, just a modified protocol to include barcodes into the RNA prep. So as you can see on the right, so the prep is pretty much similar, except the reverse transcription step and the sequencing step, both adapters at this point are RNA, not DNA, and that allows you to the motor to go through seamlessly, and this gives us nice barcoding results. So as you can see on the plot on left, this is the percentage of reads from barcoding. So the barcoding balance is quite nice. So so we are releasing 24 barcodes. So you expect about 4% average coverage, and that's what you see. So the coverage of barcoding is quite nice. So with the barcoding kit, we also want to make sure that you get all the other features that we talked about in RNA-004. So on left, what you see is the 4-plex readout over time, similar to simplex RNA. So millions of reads is very similar to normal RNA-004 kit. You get about like 15 million reads. And also with the barcoding kit in the middle, you see that you get poly A tail length right as well. And then on right, what you see is the correlation of barcoding classification. So all 24 barcodes, you can identify them accurately from each other. This barcoding kit is also the features are available in Dorado, and the barcoding kit is already in beta testing as well. And we have some of the press data coming in, so this week from Methane. Thank Methane. And so this shows like when you use our barcoding kit, and the number of reads he gets is about 25 million. It's pretty similar to what we get from RNA-004 kit. And on the right-hand plot, what you see is where he did 5 samples, mRNA, 4 mRNA samples, and 1 RNA-CS, barcoded, run on the same flow cell, and you can see the differences in read length versus density. So RNA-004 barcoding kit is now in beta testing, and hopefully that will be very useful in the RNA community. So RNA modifications, as I said, we are the only technology that can detect RNA modifications. We are the only technology that can pass RNA directly through the nanopore. So these 4 modifications, M6A, inosine, M5C, and pseudouridine, they are already available with our in Dorado with our modification calling. What we have done recently is we have improved the accuracies of these modifications even further. So by doing so, we have reduced the false positive. What you see on the in each of these confusion matrices, the top right is the false positive, and the number is now quite low, which means the accuracy has already been increased. So you can see on the plot on right, so these are synthetic IVT molecules with modifications imprinted in specific places, and you can see we can call them accurately where the modification should be. So we are now introducing 4 more modifications to RNA. This is RNA 2' or methyl. So 2' or methyl modification is in the sugars. Most of the other modifications are actually on the base. So why this is important? Because 2' or methyl modifications are critical. They have critical biological roles for stability, folding, translation, and immune recognition. They are very abundant in ribosomal RNA, and they are involved in pathologies, and they are used very much in RNA therapeutics as well, especially in guide RNA with the CRISPR technology. 2' or methyl is an important modification. So we are now introducing 4 modifications, 2' or methyl A, C, G, and U. So again, on left, what you see is the dark bar shows where the modification should be, and you can see in all the control strands, we can call these modifications very accurately where it should be. And on right, what you see is the confusion matrices numbers. Again, false positive are really low, and the accuracies are really high, around 99%. So this is one example to show RNA modification calling. Also, I should mention that all these modifications, all 8 modifications can call simultaneously. You don't have to do one by one as well. So this is a real example of ribosomal RNA, 2 different fragments from 18S and 28S. When you call with the previous version of the version 5.1, which only had 4 modifications, M6A, inosine, M5C, and pseudouridine. You can see the modification calling. And now when you call it with all 8, with the additional 4 modifications, 2' or methyl A, C, G, and U, you can see how nicely these modifications come along. And you can also see how abundant they are in ribosomal RNA. So this is really big improvement for modification calling. Again, I highlight that we are the only technology that can detect these RNA modifications. Now, moving towards biopharma. So biopharma is obviously very important to us, and we are doing a few things with the biopharma industry at the moment. And one thing is the mRNA vaccine QC manufacturing. So in the mRNA vaccine manufacturing industry, they need to, when they manufacture batch, to do the QC, they need to do 3 tests mainly, identity, integrity, and purity. And then there are critical quality attributes under each, and to do that, currently they need these traditional methods. They need a multitude of different technologies which require quite a lot of resources, and it takes time, and all these things can be replaced by a single nanopore sequencing direct RNA test. So we have also introduced plasmid sequencing test as well. That's part of this package, and this can be done with Q-line, and this is now, so we are working with quite a lot of biopharma, and this is, this workflow is available with commercial agreements at the moment. So in this workflow, like there's the first part of the workflow is operator hands-on. So you first do the sample prep, and then user define the criteria, what is acceptable criteria for the experiment, and then you load the flow cell and the run starts, and then epitome automation takes over, and you do the rest is done by the epitome workflow. So it will generate data depending on the criteria you set, whether it is time or the number of reads you need. And then once the data collection is completed, analysis will kick off, and then finally you get a report generated with pass and fail depending on the criteria you set up, and it will give information about canonical basis modifications, identity, poly-A tail, integrity, consensus Q-score, and so on. So this workflow is now available for biopharma as well. And beyond mRNA vaccine QC, we obviously want to do other things with other things in biopharma. One of the things we are doing is this adventitious viral agents QC test. So adventitious agents are contamination that you see in biological samples, whether it is microbiological, bacteria, viral, or any other things. So this AVA is particularly focused on viral part. To do that currently, biopharma industry have to again use multitude of different technologies, and they need to do a tissue culture test, and they need to do animal modeling to do antibody testing, which require quite a lot of different instrumentations as well. And all those things, again, can be replaced with a simple nanopore reading sequencing experiment. And we, again, have Q-line. We are developing a proper workflow for AVA QC test as well. And now I want to move focus shift again slightly to talk about something very exciting. Expanding horizons into proteomics. So this is the last slide of my talk in last year NCM in Boston. So I promised that in London Calling 2025, I will talk about proteomics. So keeping my promise, so I want to talk a bit more about the proteomics and what we have developed in this area. So proteomics is obviously the vast space, and as you know, like, again, different version of the central dogma of biology. In human genome, there are about 23,000 genes, and they will tell you what could happen in your body. They transcribed into RNA. There are about 100,000 different transcript isoforms, and then they translate into proteins. And there are more than 1 million proteins in human body, and with more than 400 post-translational modifications. And they will tell you what is actually happening in your body. So the functional layer of biology is protein, if the DNA is the blueprint. So the proteins will tell you the health and disease status of your body. They are the markers of gene expression and regulation, and they are the primary targets for drugs, and obviously then post-translational modifications matters. So why protein is important in different fields? Like in research, you need to understand biology to unravel disease mechanism, find novel biomarkers, and then you can use that knowledge in biopharma to discover new therapies, determine mechanisms of action, and develop diagnostic tests. In clinical diagnostics, obviously, best real-time indicator of diseases are proteins, and in applied and industrial settings, you use protein detection in many areas, including biosecurity detection, better biologics QC, and food safety, and so on. So proteomics is very challenging. Like, if you think like DNA and RNA is challenging enough, welcome to proteomics. So proteomics are I'm listing like few challenges that we have. So one of the main challenges is the dynamic range and the sensitivity. So if you take human proteome, there are some proteins that have only like 10 copies, and some proteins, there are 10 million copies. So you need to be able to detect everything in between, and we don't have amplification for proteins like you have for DNA and RNA with PCR. And then to make things worse, methods. There are about like more than 400 different post-translational modifications, and most methods in proteomics cannot detect these post-translational modifications accurately, or at least in sequence. And also proteomics is difficult to quantify, and the reproducibility is low, and also most of the methods available for proteomics cost is very high. So the most common method for proteomics at the moment is mass spectrometry, as you know, and all these challenges are common for mass spectrometry as well. The main challenge is actually the dynamic range. So you cannot, even the mass spec cannot detect very, very low abundant proteins. And if you go towards that range, then on the other side, the cost of mass spectrometry will go really high. So the most kind of high spec mass specs are about like more than $1 million in cost. So this is where nanopore sequencing or nanopore sensing technology comes to play. So with nanopore, obviously we have a very modular platform, and we can switch component very easily without having to redesign the entire system. So you can take the nanopore out from DNA or RNA flow cells and introduce a new nanopore in for protein. And then we also have different methods to do sample prep. So if you take mass specs out, so the other most common methods to do proteomics at the moment currently is indirect detection of proteins. What I mean by indirect is you take the protein of interest, which is shown in yellow, and then people use either antibodies or aptamers, bind to them, and then you use the binding molecule, which is mostly DNA, as the proxy to measure the presence and absence of proteins or quantify the proteins. So the DNA is the readout. Obviously, being DNA sequencing company, we can do that as well. What we want to do is going to more direct protein analysis. So in the direct protein analysis, what we are trying to do is pass the DNA, pass the protein molecule directly through the nanopore, so you get actually the readout from the protein, not from a proxy molecule, and it provides information about post-translational modification, like we get a DNA and RNA modification in that field. So we have So we are developing two different methods to be able to do that. So one is peptide method, and the second one is full-length method. So in peptide method, this is a video to show what we currently do. This is our workflow. So you take the peptide of interest, and you encapture that peptide in between two DNA handles. And then we use this DNA handle with the DNA motor we currently use. So that will bring that analyte back to the nanopore. So the nanopore will read the DNA part first. So you get the DNA signal. In between the DNA and peptide, we have a polynucleotide region. That's why you see a flat signal. This is to separate the molecule protein. And you see the protein peptide signal in yellow. And then again, you see the flat line, which is the polynucleotide region. So we can do that in a very free-range system and using DNA motor to control the movement. So we can get peptide after peptide after peptide in a single line, in a single experiment. And then we have developed this method into two different applications. The first one is protein biomarker detection, and the second one is protein barcoding method for life sciences research tools. So this is to give an overview of what we are trying to do in proteomics space. This is our roadmap for full proteomics. In development, we have protein barcoding method, which can screen protein variants. And then we have protein biomarker detection methods, gives absolute protein quantification. And in research, we have full-length protein identification. This is from intact single molecule identification and detect protein PTMs as well. And then in the future, we are going to do towards de novo sequencing, which is actually sequencing the amino acid composition of the polynucleotide. And in between, we are also working on the affinity methods to get the DNA readout from protein either by antibody or aptamers as well. So let me walk through the protein biomarker detection and quantification method first. So we are working with a company called Genome on this particular approach to develop this method. So in this method, so as you can see on right, we have the proteome, and then we can digest the proteome into peptides. And at this stage, we add internal standards. So internal standards are basically to quantify the relative abundance of proteins. And if you don't, and then we can take this peptide pool and we can convert that into a library, as I mentioned earlier, by capturing them in between two DNA handles, and we can read through the nanopore. But if we don't enrich the peptide of interest, what you read most of the time is the most abundant proteins in human body, which is like more than 60% it is albumin. So to get rid of that problem, we use Genome antibodies, and what they do is they enrich a particular peptide per protein very accurately, and they have developed these antibodies. They are high sensitive because they have multi-epitope antibodies, so we can use those antibodies to enrich particular peptides, so we get rid of all the non-interested or highly abundant proteins, and then we use those peptides of interest in our sample prep, and then we can do the analysis of those peptides. So the advantages of this method is because we do the digestion, it gets rid of all the protein-protein interactions, so it improves specificity and sensitivity, and also because we add internal standards, we can do absolute quantification. So we add these internal standards in known concentrations, so we can get the actual concentration of the peptides, so the proteins in your sample, the protein of interest. Also with the internal standards and the very specific or high sensitivity, high specificity antibodies, we can do something called stoichiometric flattening, and that will address the dynamic range problem. So in the same sample, if you have molecules of like 10 molecules or 10 million molecules, we can detect them all and get the actual quantification as well. And because we use DNA handle, we can barcode the DNA, and the nanopore we use in this particular application not only can read the peptide, but also can read the DNA to more than Q20 accuracy, which means we can barcode the DNA and we can read that barcode to multiplex protein samples. So we can barcode not only the different peptides, but we can also barcode different samples into same flow cell, which reduces the cost significantly. So, so this is one example that I'm going to show you. So we picked up 12 biomarkers from the menu of the Genome, and these biomarkers come from oncology, cardiovascular, and inflammation panels. So then what we did was we synthetically generated peptides for those biomarkers, and we created a classification method. So in this classifier, so we can we generate data, and then we build the classifier to identify these peptides. So on the left side plot, what you see is this classification of these peptides. So as you can see, the predicted versus true correlation is really high, which means we can very accurately detect these peptides separate from each other. And we have achieved 99.4% 12-way accuracy, and all peptides above 99% accuracy. And we have also included the background peptides. This is background peptides are coming from HeLa cells, and we can not only separate peptides from each other, these 12 peptides, but we can separate them from background as well. So this is a really nice experiment to see. And more importantly, the most exciting part is the sensitivity that we can go to. So in this experiment, what I'm showing is the on x-axis actual flow cell input, and y-axis measured input. So this is done with PSA biomarker. So PSA is the main biomarker for prostate cancer. So we have synthesized this biomarker, and then we have generated the library with this biomarker, and then we purified this library to very high purity, and then we added this library library into the flow cell in in known concentrations. So that's what you see on x-axis. In y-axis, what you see is what you actually read. So you can see the correlation is really, really high. So in the dotted line is y equals x, and so when we showed this data actually to mass spec specialists, and they were really surprised to see that we can go to this 100 septomolar level of detection limit. So that sensitivity is something that you don't really see. And in mass spec, you usually, even very high-end mass spec, you go towards 1 attomolar level sensitivity, and which means we are now introducing a new term into proteomics, femtomole level of sensitivity detection. So this is very exciting, and most of the mass spec specialists that we spoke to are very excited about this result as well. And so which means with this particular application, so we are showing ultra-low detection limit, which enables detection of biomarkers at a level previously unattainable, and wide dynamic range enables measurement of panels of biomarkers with wide dynamic range in complex mixtures. Ultra-high sensitivity enables more frequent monitoring of patient samples and analysis of hard-to-obtain samples. High precision means you provide absolute biomarker quantification, and multiplexing both at the biomarker level and at the sample level means you can reach unparalleled sample throughput, which lowers the cost significantly. So the second application that we are developing with this protein sensing is the protein barcoding as a life science research tool. So this is an application mainly for protein purification. So what we do is we purify proteins with barcodes intact, and then we release those barcoding tags, and then we can encapsulate those barcodes in between two DNA handles to do the library prep, and then we can read the barcode. By doing so, we can identify the protein of interest. So example use cases of this is like protein expression, relative protein abundance, protein binding. Imagine there's a situation that because we are now in AI era using all these machine learning AI tools, people design thousands and thousands of protein variants. Assume you design 1,000 enzymes, enzyme variants, and you need to know which mutant or which the top 10% of the top binders for a particular assay. So in that case, you can do these 1,000 different enzyme variants. You can purify them with these particular barcodes, and you can do the binding assay, and at that point you can release the barcodes, and you can do the library prep with nanopores, and you can identify what are those active enzyme variants are. So, so we have data for that as well. So in this particular example, we have done 20 barcodes. So we synthesized 20 barcodes, and then we generated the classifier. And then in this example, what I'm showing on left is the correlation of these 20 barcodes, how nicely we can separate them and identify them in this mixture of 20 targets. So accuracies, the mean accuracy is 98.9 for all barcodes. And we want to go beyond 20 to be able to do thousands of barcodes. So we have done a 1,000-way classifier. So we have built a classifier, and then within that 1,000, we want to see whether we can select 20 barcodes, a small subset, accurately. So what this shows is, in this 1,000 classifier, we can accurately select the 20 barcodes that we wanted to select. So which means this method is very scalable, and we want to go beyond 1,000 as well. So we will develop more methods to do that. And also, not only this method can detect the presence and absence of the barcode, it can also do the relative abundance quantification. So in this example, what we did was, within those 20 barcodes that I showed in the previous slide, we chose 4 barcodes, and we used them in one protein, and we expressed those 4 proteins with the same protein with 4 different barcodes, and then we mixed them in different quantities. So on the plot on the right, on the left, what you see is 1 to 1 to 1 to 1 ratio. So when we release those barcodes from the mixture and try to detect the presence and absence of the barcode and the relative abundance, we get really nicely identify the 4 barcodes that we put into the system. So those are the barcodes on towards the left of the dotted line. Towards the right of the dotted line is the barcodes that we didn't put, so you don't see them. And also, importantly, you see the 1 to 1 to 1 to 1 ratio. In the other example, the ratio is 100 to 10 to 10 to 1, and you can see again very nicely, we can detect those relative abundance of the barcodes in the sample. So not only the barcode and relative abundance and the identifications, post-translational modification is very, very important in proteins. As I said before, like we do in DNA and RNA, in proteins we can do protein identification through nanopore. So what this shows is the 3 different modifications: phosphorylation, succinylation, and acetylation. And on the right plot, you can see the accuracies on this matrix. Accuracies are relatively high, and even in the signal space, you can see the difference of the PTM modification. So you can, on the bottom panel, what you see is the black line is the unmodified signal, and when it is modified with phosphorylation or succinylation or acetylation, you can see how much the signal changes, and this is single molecule level experiment, so you can clearly see the signal difference, and we can detect that with our machine learning AI tools as well. So as the summary, so we have, I have shown that we have 2 different applications: protein biomarker detection and protein barcoding. And then we want to go towards full-length protein translocation as well. So in this particular example, the signal in the full-length transcript box is coming from about 300 amino acid protein, and also there are about 150 signals overlaid on top of each other to generate that signal. So you can see the reproducibility is quite high. Jeff Nivala will talk about this approach more, and the last talk tomorrow is from Jeff generally about this method, how do we develop a nanopore sensing method for full-length translocation of proteins. So again, going back to Apple, iPhone, iPad, and Apple Watch. So as I showcase, I think we can do all these different applications on the same set of devices. So whether it is DNA, RNA, or protein, you can do it on MinKNOW, you can do it on GargantION, you can do it in Promethean. And so we have this ecosystem that is quite nicely, we can do anything, anywhere, by anyone. So this is our theme. So I have shown that we can do genomics and epigenomics, and that tells you what could happen in your body. And then transcriptomics and epitranscriptomics tell you what is being planned in your body. And then proteomics, I showed that the new applications that we are developing, and hopefully we will commercialize this soon as well. So in the previous slide, I forgot to say that there was, if I go back, so register your interest if you are interested in protein sensing applications with Oxford Nanopore. So protein early access research labs, like which is Pearl now. So please do let us know if you are interested, and we are very happy to work with the community and to develop proteomic applications as well. So genomics, transcriptomics, and proteomics. So proteomics and post-translational modifications will tell you what is happening in your body, but then to connect all these dots, you need a metabolomics as well. So metabolomics is something new, but with metabolomics, if you connect all dots, you will see with everything else what has really happened in your body. Maybe that will be my next promise for next year London Calling. So last thing is the timelines, obviously very important. So I will invite Rosemary to talk about timelines as well. So thank you very much.
Thanks, Akmal, and thanks to everyone. So we see what we've delivered already, so gone live by the talent of our amazing ML group has been Dorado V1 during or just before this talk. And then, of course, all of the sort of 10x work that's happened. We have an enormous amount of end-to-end workflows going out. So telomere sequencing went out, chromatin sequencing went out. We will have PGX, hereditary cancer, among other things going out this year. And then on the software and algorithm side, I made a special box to say raw data off by default, just to remind everyone, because we know what happens if we don't remind him. And then platform chemistry, protein registration of interest, gone live now. Please do put your thoughts down on paper. The MinKNOW Access program, we got thousands of applications for that, and we are still delivering on the asks of people who registered for the MinKNOW Access program. You have no idea how powerful it is to understand your wants and needs for a new technology as you develop it. And then, of course, we have high-output chemistries, RNA barcoding, the 16S multiplexing all going out as well. We have an enormous amount of work going on the hardware side that I haven't spoken about today, but we'll talk about tomorrow morning around our DX instrumentation, Elysian GridION. So, yeah, another year at Nanopore.